Hi scientists! Today we're going to talk about the process of mitosis. Our big idea for this lesson is that when cells divide, each daughter cell ends up with a copy of the parent cell's DNA. Cells go through a very specific process to make sure that each cell ends up with that set of DNA, so the new cells have the directions that they need to do their jobs and exist. Before we jump into the process, it's important to talk about a little bit of vocab. Some of this will be review and some will be new. When we talk about DNA, we talk about two different forms. First, we have chromatin, which is uncondensed or loose DNA. It's important that the cell can open up the DNA to be able to read the code and use it. So most of the time, the cell, or during interphase, the cell has DNA as chromatin. When it's time for the cell to divide, the DNA condenses or packs into structures called chromosomes. One way that I like to think about this is about packing for a trip. When you're home, it's okay if your clothes and things are a little bit all over the place. But when it's time to pack for a trip, you want to fold everything nicely and put it in your suitcase and pack it all really nicely. It would be a disaster if you just had an armful of clothes and you walked onto an airplane with an armful of clothes, right? You would have a hard time keeping track of it. You'd probably lose some stuff. But having everything all nicely packed in your suitcase means that nothing's going to get lost, hopefully, and that it'll all be just as it was when you get to your destination. So here we have the same kind of thing. We have loose, all over the place DNA that's totally fine and great for when our cell is just doing its job. But when it's time for the cell to divide, we want to make sure that everything is packed up nice and tight and organized so we can keep track of it and everything ends up at its destination, safe and sound. You'll notice that this chromosome is represented as an X, which is often how we draw them. We talk about the two sides of this X, and we call each side a sister chromatin. And so the two sides are identical. In S phase, we replicated our DNA, and so now we have two sets of identical DNA. One of these sister chromatids, one set of DNA, is going to end up in one of the daughter cells, and the other will end up in the other daughter cell. So during mitosis, this chromosome will split down the middle. The two sister chromatids are held together with a structure called a centromere, and I've represented this with a little circle. And so the centromere holds those sister chromatids together, and it also provides an attachment point for structures called spindle fibers. I'm going to introduce spindle fibers here, but they'll make more sense when we talk about the process of mitosis. So spindle fibers are protein structures, and they are going to anchor the chromosomes during mitosis. Now we'll talk through the stages of mitosis. We have 4 stages: prophase, metaphase, anaphase, and telophase. So prophase comes first. I like to think about the beginning of this as being like pro or pre, so it's first, it's where we're really getting ready. So here our chromatin condenses into chromosomes, right? Our DNA gets all packed up and ready to go, and our nuclear membrane dissolves. So you can see in the drawing that I have here, we no longer have a nucleus, we just have chromosomes sort of floating in the cell. And then our spindle fibers start to form. You can see here I've also drawn in these star-shaped structures. They are called centrioles, and they act like the poles of the cell, like the south pole and the north pole. And they're going to be where some things attach and be kind of the anchor points for the new cells. After we have our chromosomes condensed, the next step is that they'll line up in the middle of the cell. So this stage is called metaphase. I like to think M and middle, and that's how I remember what happens here. And so you can see that they are lined up in a nice straight line. We have our spindle fibers attached to the centromeres and then going all the way to the centrioles, right, to the poles of the cell. Next we have anaphase. I like to think about the A in anaphase as being the same as the A in apart, because here we see that the centromeres break and the two sides that are sister chromatids are being pulled to opposite ends of the cell along those spindle fibers. We also sometimes start to see that the cell pinches in here because cytokinesis is starting or happening at the same time. And then finally, we have telophase, where our sister chromatids, right, our identical sets of DNA, are in the space that they're going to need to be when the cell divides, and our nuclear envelope, our nuclear membrane, is starting to reform. And then our chromosomes will decondense, they'll go back into chromatin, so that that DNA, that code, is ready to be read and help the new cell do its job. So again, as a quick review, in prophase, we start to see our chromosomes form and our machinery is getting ready for the process of mitosis. In metaphase, the chromosomes line up in the center of the cell. In anaphase, they're pulled apart towards the opposite poles of the cell. And then in telophase, our nucleus starts to come back together and our chromosomes decondense back to chromatin. You'll see the acronym I have at the bottom here is PMAT, and that's how many of us remember these stages. So I encourage you to do the same. If you have any questions, please leave them in the comments or stop by office hours.